So hello dear learners, today we are going to study unit 4, that is plant physiology. In this unit there are 3 chapters: photosynthesis in higher plants, respiration in plants, plant growth and development. So today we are going to study photosynthesis in higher plants. The description of structure and variation of living organism over a period of time ended up as two apparently irreconcilable perspectives on biology. The two perspectives essentially rested on two levels of organization of life forms and phenomena. One described at organismic and above level of organization, while the second described at cellular and molecular level of organization. The first resulted in ecology and related disciplines, the second resulted in physiology and biochemistry. Description of physiological processes in flowering plants as an example is what is given in the chapters in this unit. The processes of photosynthesis, respiration and ultimately plant growth and development are described in molecular terms, but it in the context of cellular activities and even at organism level wherever appropriate. The relation of the physiological process to environment is also discussed. Melvin Calvin, born in Minnesota in April 1911, received his PhD in chemistry from the University of Minnesota. He served as professor of chemistry at the University of California, Berkeley. Just after World War II, when the world was under shock after Hiroshima and Nagasaki bombings, and seeing the ill effects of radioactivity, Calvin and co-workers put radioactivity to beneficial use. He, along with J. Basham, studies reaction in green plants forming sugar and other substances from raw material like carbon dioxide, water and minerals by labeling the carbon dioxide with C14. Calvin proposed that plants change light energy to chemical energy by transferring an electron in an organized array of pigment molecules and other substances. The mapping of the pathway of carbon assimilation in photosynthesis earned him Nobel Prize in 1961. The principles of photosynthesis as established by Calvin are at present being used in studies on renewable resources for energy and materials and basic studies in solar energy research. Chapter 11, that is photosynthesis in higher plants. Read with us because reading is first step toward a good learning. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plants make their or rather synthesize the food they need for photosynthesis and are therefore called autotrophs. You have already learned that the autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are heterotrophs. Green plants carry out photosynthesis, a physico-chemical process by which they use light energy to drive the synthesis of organic compounds. Ultimately, all living forms on earth depend on sunlight for energy. The use of energy from sunlight by plant doing photosynthesis is the basis of life on earth. The photosynthesis is important due to two reasons. It is the primary source of all food on earth. It is also responsible for the release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? In this chapter focuses on the structure of photosynthetic machinery and the various reactions that transform light energy into chemical energy. What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in the earlier classes have shown that chlorophyll, green pigment of leaf, light and CO2 are required for photosynthesis to occur. You may have carried out the experiments to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with the black paper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of light. Another experiment you may have carried out where a part of leaf is enclosed in a test tube containing some KOH soaked cotton while the other half is exposed to air. The setup is then placed in light for some time. On testing for the presence of starch later in the two parts of the leaf, you must have found that the exposed part of the leaf tested positive for starch while the portion that was in the tube tested negative. This showed the CO2 was required for the photosynthesis. Can you explain how this conclusion could be drawn? Early experiments. It is interesting to learn about those simple experiments that led to a gradual development in our understandings of photosynthesis. Joseph Priestley in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plants. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning on a closed space soon gets extinguished. Similarly, a mouse would soon suffocate in a closed space. He concluded that burning candle or an animal that breathes in air both somehow damage the air. But when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plants restore the air whatever breathing animals and burning candles remove. Can you imagine how Priestley would have conducted the experiment using the candle and plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley, but by placing it once in the dark and once in the sunlight, Jan Ingenhouse showed that sunlight is essential for the plant's process that somehow purifies the air fouled by burning candles or breathing animals. Ingenhouse, in an elegant experiment with an aquatic plant, showed that in bright sunlight, small bubbles were formed around the green plant parts, while in dark they did not. Later, he identified these bubbles to be of oxygen. Hence, he showed that if it is only the green plant that could release oxygen. It was until about 1854 that Julius von Sachs provided evidence for production of glucose when plants grow. Glucose is usually stored as starch. His latest study showed that the green substance in plants, chlorophyll as we know it now, is located in a special body later called chloroplast within the plant cell. He found that the green parts in plants is where glucose is made and that the glucose is usually stored as starch. Now, consider the interesting experiment done by T.W. Angry Man. Using a prism, he split light into its spectral components and then illuminated a green alga, cladophora, placed in a suspension of aerobic bacteria. The bacteria were used to detect the sites of photoevolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. The first action spectrum of photosynthesis was thus described. It resembles roughly the absorption of spectra of chlorophyll A and B. By the middle of the 19th century, the key features of plant photosynthesis were known, namely that plants could use light energy to make carbohydrates from CO2 and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was then understood as CO2 plus H2O equals light. CO2 plus H2O and light gives CH2O plus O2, where CH2O represents a carbohydrate, glucose a 6 carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by a microbiologist Cornelius Van Niel, who, based on his studies of purple and green bacteria, demonstrated that photosynthesis is only a light-dependent reaction in which a hydrogen forms a suitable oxidizable compound, reduces carbon dioxide to carbohydrates. This can be expressed by 2H2A plus CO2 and light 2A plus CH2O plus H2O. In green plants, H2O is the hydrogen donor and is and is oxidized to O2. Some organisms do not release O2 during photosynthesis. When H2S instead is the hydrogen donor for purple and green sulfur bacteria. The oxidation product is sulfur or sulfate depending on the organism and not O2. Hence, he inferred that the O2 evolved by the green plant comes from H2O, not from carbon dioxide. This was later proved by using radioisotopic technique. The correct equation that would represent the overall process of photosynthesis is therefore 6CO2 plus 12H2O plus light, 6 C6H12O6 plus 6CO2, 6O2 and 6H2O. Where C6H12O6 represents glucose, O2 released in the form of water. This was proved using radioisotope techniques. Note that this is not a single Reaction but description of multi-step process called photosynthesis. Can you explain why 12 molecules of water as substrate are used in the equation given above? See the equation and answer this question. Where does photosynthesis take place? You would of course answer in the green leaf or in the chloroplasts. Based on what you earlier read in chapter 8, you are definitely right. Photosynthesis does take place in the green leaves of plants, but it does also in other green parts of the plant. Can you name some other parts where you think photosynthesis may occur? You would recollect from previous unit that the mesophyll cells in the leaves have a large number of chloroplasts. Usually the chloroplasts align themselves along the walls of mesophyll cells such that they get optimum quantity of the incident light. When do you think that the chloroplast will be aligned with their flat surface parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast, there is membranous system consisting of grana, the stroma, lamella, and the matrix stroma. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn forms starch. The former set of reactions, since they are directly light driven, are called light reaction. Photochemical reaction, the latter are not directly light driven but are dependent on the products of light reactions, ATP and NADPH. Hence, to distinguish the latter, they are called by convention as dark reaction. Carbon reaction, however, they should not be constructed to mean that they occur in darkness or that they are not light dependent. This is a structure of chloroplast. It is the outer membrane, it is the inner membrane. See there. This black layer, this layer is outer membrane, this layer is inner membrane, and this is stroma, lamella, these are grana, and they all made thylakoid. This is starch granule, lipid droplet, and ribosomes. Stroma, the liquid is stroma. How many types of pigments are involved in photosynthesis? Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves, even in the same plant? We can look for an answer to this question by trying to separate the leaf pigments of any green plant through paper chromatography. A chromatographic separation of the leaf pigment shows that the color that we see in the leaves is not due to a single pigment, but due to 4 pigments: chlorophyll A, bright or blue-green algae, blue-green in the chromatograph; chlorophyll B, yellow-green; xanthophylls, yellow; and carotenoids, yellow to yellow-orange. Let us now see what roles various pigments play in the photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelengths. Can you guess which is the most abundant plant pigment in the world? Let us study the graphs showing the ability of chlorophyll A pigment to absorb lights of different wavelengths. Figure 11.3a. This diagram. Pigments are substances that have an ability to. Of course, you are familiar with the wavelength of visible spectrum of light as well as Vibgyor. These are colors of a light spectrum, and we also say that these are colors of a rainbow. From figure 11.3a, can you determine the wavelength color of light at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelength too? If yes, which one? See this diagram and answer the given questions. Now look at figure 11.3b showing the wavelengths at which maximum photosynthesis occurs in plant. Can you see that the wavelengths at which there is maximum absorption by chlorophyll A, it means in the blue and the red regions, also shows higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 11.3c, can you say that there is a complete 1-to-1 overlap between the absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? This is diagram 11.3c. These graphs together so that most of the photosynthesis takes place in the blue and red regions of the spectrum. Some photosynthesis does take place at the other wavelength of the visual spectrum. Let us see how this happens. Through chlorophyll is the major pigment responsible for trapping light. Other thylakoid pigments like chlorophyll B, xanthophylls, and carotenoids, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelengths of incoming light to be utilized for photosynthesis but also protect chlorophyll A from photooxidation. What is light reaction? Light reactions or photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical and chemical intermediates ATP and NADPH. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes LHC within photosystem PS, photosystem 1 and photosystem 2. PS second. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LHC, that is light harvesting complexes, are made up of hundreds of pigment molecules bound to proteins. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system also called antennae. This is antennae or antenna. Figure 11.4. This is the diagram of this. This is the diagram. Let us study out. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystem. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nm, hence it is called P700, while in PS2 it has absorption maximum at 680 nm and is called P680. The electron transport in photosystem second, the reaction center chlorophyll A absorbs 680 nm wavelength of red light, causing electron to become excited and jump into an orbit further from the atomic nucleus. These electrons are picked up by an electron acceptor, which passes them to an electron transport system consisting of cytochromes. This movement of electron is downhill in terms of oxidation, reduction, or redox potential scale. The electrons are not used up as they pass through the electron transport chain but are passed on to the pigments of photosynthesis photosystem PS1. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nm and are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy-rich NADP+. The addition of these electrons reduces NADP+ to NADP+ H+. This whole seam of transfer of electrons starting from PS2 uphill to the acceptor, down the electron transport chain to PS1, excitation of electron transfer to another acceptor, and finally downhill to NADP+, reducing it to NADP H+. H+ is called the Z scheme. Due to its characteristic shape, this shape is found when all the carriers are placed in a sequence on a redox potential scale. Then the topic is splitting of water. You would then ask, how does PS2 supply electron continuously? The electron that were removed from photosystem second must be replaced. This is achieved by electron available due to splitting of water. The splitting of water is associated with PS2. Water is split into 2H+O and electrons. This create oxygen, one of the net products of photosynthesis. The electrons needed to replace those removed from photosystem first are provided by photosystem second. The reaction is given here. 2H2O gives 4H plus O2 and 4 electrons. We need to emphasize here that the water splitting complex is associated with the PS2, which itself is physically located on the inner side of the membrane of the thylakoid. Then where are the protons and O2 formed likely to be released? In a lumen or not on the outer side of the membrane. Cyclic and non-cyclic phosphorylation. Living organisms have the capability of extracting energy from oxidizable substances and store this in form of bond energy. The substances like ATP carry this energy in their chemical bonds. The process through which ATP synthesis is synthesized by cells in mitochondria chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first PS second and then PS first, a process called non-cyclic phosphorylation occurs. The two phosphosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH plus and H plus are synthesized by this kind of electron flow. When only PS first is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electron. The possible location where this could be happening is in the stroma lamella. When the membrane or lamella of the grana have both PS first and PS second, the stroma lamella membranes lack PS second as well as NADP reductase enzyme. The excited electron does not pass on the NADP plus, but is cycled back to the PS first complex through the electron transport chain. The cyclic flow hence result only in the synthesis of ATP but not NADPH plus H plus. Cyclic photophosphorylation also occurs when the only light of wavelength beyond 680 nm are available for excitation. Chemiosmotic hypothesis. Then we will study where are the ATP and NADPH used. Then we will study the primary sector of CO2, the Calvin cycle, the C4 pathway, photorespiration, factors affecting photosynthesis, light, carbon dioxide concentration, temperature, water, and then some. Well, let us study. Let us now try to understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism. Like in respiration in photosynthesis, too, ATP synthesis is linked to development of a proton gradient across a membrane. This time these are membranes of thylakoid. There is one difference. So here the proton accumulation is toward the inside of the membrane. It means in the lumen. In respiration, protons accumulate in the intermembrane space of the mitochondria where electrons move through their ETS. Let us understand what causes the proton gradient across the membrane. We need to consider again the processes that take place during the activation of electrons and their transport to determine the steps that cause the proton gradient to develop. First, since the splitting of the water molecule take place on the inner side of the membrane, the protons and hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. B. As electrons move through the photosystem, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the other side of the membrane, transfer its electron not to an electron carrier but to an H carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into inner side of lumen or inner side of the lumen side of the membrane. The NADPH reductase enzyme is located on the stroma side of the membrane along with the electrons that come from the acceptor of electron of PS first. Protons are necessary for the reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in the number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. Why are we so interested in proton gradient? This gradient is important because it is the breakdown of gradient that leads to the synthesis of ATP. The gradient is broken down due to the movement of proton across membrane to the stroma through the transmembrane channel of CF 0 to the of the ATP synthase. The ATP synthase enzyme consists of two parts. One called CF0 is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called CF1 and protrudes on the other surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which make the enzyme synthesis several molecules of energy-packed ATP. Chemioosmosis requires a membrane, a proton pump, a proton gradient, and ATP synthase. Energy is used to pump protons across a membrane to create a gradient or a high concentration of protons within the thylakoid lumen. ATP synthase has a channel that allows diffusion of protons back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyze the formation of ATP along with the NADPH produced by the movement of electrons. The ATP will be used immediately in the biosynthesis reactions taking place in the stroma responsible for fixing CO2 and synthesis of sugars. Where are the ATP and NADPH used? We learned that the products of light reaction are ATP, NADPH and O2. Of these, O2 diffuses out of the chloroplast, while ATP and NADPH are used to drive the processes leading to the synthesis of food. More accurately, sugars. This is the biosynthetic phase of photosynthesis. This process does not directly depend on the presence of light but is dependent on the products of light reaction. It means ATP and NADPH. Besides CO2 and H2O, you may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic processes continue for some time and then stops. Then light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Discuss the amongst ourselves. Let us now see how the ATP and NADPH are used in biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce CH2O or sugars. It was the interest of scientists to find out how this reaction proceeded, or rather, what was the first product formed when CO2 is taken into a reaction or fixed just after the World War II. Among the several effects of to put radioisotopes to beneficial use, the work of Melvin Calvin is exemplary. The use of radioactive C14 by him in algal photosynthesis studies led the discovery that the first CO2 fixation product was 3-carbon organic acid. He also contributed to the working of complete biosynthetic pathway, hence it was called Calvin cycle after him. The first product identified was the 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also try to know whether all plants in PGA has the first product of CO2 fixation or whether any other product was formed in other plants. Experiment conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of CO2 fixation was again an organic acid, but one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid, or OAA. Since then, CO2 assimilation during the photosynthesis was said to be two main types. Those plants in which the first product of CO2 fixation is C3 acid PGA, the C3 pathway, and those in which the first product was C4 acid OAA, it means the C4 pathway. These two groups of the plants showed the other associated characteristics that we will discuss later. The primary acceptor of CO2. Let us now ask ourselves a question that was asked by scientists who were struggling to understand the dark reaction. How many carbon atom would have molecule have which after accepting fixing CO2 would have 3 carbons of PGA? The studies very unexpectedly showed that the acceptor of molecule was a 5-carbon ketose sugar ribulose bisphosphate RUBP. Did any of you think this possibility? Don't worry. The scientists also took a long time and conducted many experiments to reach this conclusion. They also believe that since the first product was C3 acid, the primary acceptor would be 2-carbon compound. They spent many years trying to identify a 2-carbon compound before they discovered the 5-carbon RUBP. The Calvin cycle. Calvin is co-workers and work out with the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Calvin pathway operates and where the sugars is synthesized. Let us at the outset understand very clearly the Calvin pathway occurs in all photosynthetic plants. It does not matter whether they have C3 or C4 or any other pathways. For ease of understanding, the Calvin cycle can be described under 3 stages: carboxylation, reduction, and regeneration. Carboxylation. The carboxylation is the first of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of Calvin cycle, where CO2 is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP or carboxylase, which results in the formation of two molecules of 3-PGA. Since the enzyme is also has an oxygenation activity, it would be more correct to call RUBP carboxylase oxygenase or RUBASCO. This is the cycle. Reduction. These are the series of reactions that led the formation of glucose. This step involves utilization of 2 molecules of ATP for phosphorylation of 2 NADPH after reduction per CO2 molecule. The fixation of 6 molecules of CO2 and 6 turns of cycle are required for the formation of 1 molecule of glucose from the pathway. Regeneration of CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP. Hence, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 of NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in dark reaction that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of the cycle are required. Work out how many ATP and NADPH molecules will be required to make 1 molecule of glucose for the Calvin pathway. It might help to understand all of this. We took at what goes and what comes out of the Calvin cycle. N 6 CO2 out, 1 glucose, and 18 ATP out, 18 ADP in, 12 NADPH out, 12 NADP. The C4 pathway. Plants that are adapted tropical regions have the C4 pathway mentioned earlier. Though these plants have the C4 oxaloacetic acid, the first CO2 fixation product that they use, the C3 pathway or the Calvin cycle. As for the main biosynthetic pathway, then what way are they different from C3 plants? This is a question that you may reasonably ask. C4 plants are special. They have a special type of leaf anatomy. They tolerate higher temperatures. They are responsible to highlight intensities. They lack a process called photorespiration and have greater productivity of biomass. Let us understand these one by one. Study vertical section of leaves, one of C3 plant and another for C4 plant. Do you notice the differences? Do both have the same types of mesophils? Do they have similar cells around the vascular bundle sheath? The particularly large cells around the vascular bundles of C4 plants are called bundle sheath cells, and the leaves which have such anatomy are called crans anatomy. Crans means the breadth, and there is a reflection of arrangement of cells. The bundle sheath cells may form several layers around the vascular bundle. They are characterized by having a large number of chloroplasts, thick walls, impervious to gaseous exchange, and no intercellular spaces. You may cut a section of leaves, C4 plants, maize, or sorghum to observe the crans anatomy and the distribution of mesophil cells. It would be interesting for you to collect leaves of diverse species of plants around you and cut vertical sections of the leaves. Observe under the microscope, look for the bundle sheath around the vascular bundles. The presence of bundle sheath would help you to identify the C4 plants. Now study the pathway shown in figure 11.9. This pathway that has been named as the Hatch and Slack pathway is again a cyclic process. Let us study the pathway by listening these steps. The primary CO2 acceptor is a 3-carbon molecule, phosphonopyruvate PEP, and is present in the mesophil cells. The enzyme is responsible for this fixation, PEP carboxylase or PEP case. It is important to register that the mesophil cells lack RUBISCO enzyme. The C4 acid OAA is formed the mesophil cells and then forms other 4-carbon compounds like malic acid or aspartic acid in the mesophil cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, C4 acids are broken down to release CO2 and 3-carbon molecule. The 3-carbon molecule is transported back to the mesophil where it is converted to PEP again, thus completing the cycle. The CO2 released in the bundle sheath cell enters the C3 or Calvin pathway, a pathway common to all plants. The bundle sheath cells are rich in enzyme ribulose, biphosphate, carboxylase, oxygenase, RUBISCO, but lack PEP case. Thus, the basic pathway that results in the formation of sugars, the Calvin pathway, is common to C3 and C4 plants. Did you note that Calvin pathway occurs in all the mesophil cells in the C3 plants? In the C4 plants, it does not take place in the mesophil cells, but does so only the bundle sheath cells. Photorespiration. Let us try and understand one of the more processes that creates an important difference between C3 and C4 plants. Photorespiration. To understand the photorespiration, we have to know a little bit more about the first step of the Calvin pathway, the first CO2 fixation step. This is the reaction where RUBP combines with CO2 to form 2 molecules of 3-PGA that is catalyzed by RUBISCO. RUBP plus CO2 give an RUBISCO gives 2 into 3 PGA. RUBISCO, that is the most abundant enzyme in the world. Do you know wonder why? Answer the question. Is characterized by the fact that its active site can bind to both CO2 and O2, hence the name. Can you think how this could be possible? RuBSCO has a much greater affinity for CO2, where the O2 is nearly equal. Imagine what would happen if this were not so. This binding is competitive. It is the relative concentration of O2 and CO2 that determines which of the two will bind to enzyme. C3 in C3 plants, O2 doesn't bind to RuBSCO, and hence CO2 fixation is decreased. IUBP is instead of being converted, two molecules of PGA binds with O2 to form one molecule of phosphoglycerate and phosphoglycolate to carbon in a pathway called photorespiration. In the photorespiratory pathway, there is neither synthesis of sugars nor of ATP. Rather, it results in the release of CO2 with the utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not known yet. The C4 plant photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. This takes place when the C4 acid from the mesophyll is broken down in the bundle sheath cell in increased CO2. This results in the increasing the intercellular concentration of CO2. In turn, this ensures that the RuBSCO functions as a carboxylase, minimizing the oxidase activity. Now that you know the C4 plants lack photorespiration, you probably can understand why productivity and yields are better in these plants. In addition, these plants show tolerance to higher temperatures. Based on the above discussion, can you compare the plants showing C3 and C4 pathway? Use the table format given in table 11 by the formation. Here's the table. Factors affecting photosynthesis. Our understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of plants, including crop plants. Photosynthesis is under the influence of several factors, both internal plant and external. The plant factors include the number, size and orientation of leaves, mesophyll cells and chloroplast, internal CO2 concentration and the amount of chlorophyll. The plant or internal factors that are dependent on the genetic predisposition and the growth of the plant. The external factors would include the availability of sunlight, temperature, CO2 concentration and water. As a plant photosynthesis, all these factors will simultaneously affect its rate. Hence, the several factors interact and simultaneously affect photosynthesis or CO2 fixation. Usually, one factor is major cause or is the one of that limits the rate. Hence, at any point the rate will be determined by the factor available at suboptimal levels. When several factors affect any biochemical process, Blackman's 1905 laws of limiting factors comes into effect. This states the following: rate. If the chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. For example, despite the presence of green leaf and optimal light of CO2 condition, the plant may not photosynthesize if the temperature is very low. This leaf if given the optimal temperature, will start photosynthesizing light. We need to distinguish between the light quality, light intensity and reduction exposure to light. While discussing light as a factor that affects the photosynthesis, there is a linear relationship between incident light and CO2 fixation that rates at below light intensities. At higher light intensities, gradually the rate does not show further increase as the factors become limiting. What is interesting to note that the lighting saturation occurs at 10% of the full sunlight. Hence, except for plants and shade or in dense forest, light is really a limiting factor in the nature. Increase in incident light beyond endpoint causes the breakdown of chlorophyll and a decrease in photosynthesis. Carbon dioxide concentration. And here the summary. Carbon dioxide is a major limiting factor for photosynthesis. The concentration of CO2 is very low in the atmosphere, between 0.03 and 0.04%. Increase in concentration up to 0.05% can cause an increase in CO2 fixation rates. Beyond this, the levels can become damaging over long periods. The C3 and C4 plant respond differently for CO2 concentrations. At low light condition, neither group respond to high CO2 conditions. At high light intensities, both C3 and C4 plants show increase at the rate of photosynthesis. What is important to notice, the C4 plant source saturation is about 360 micro 1 liter per liter, while C3 responds to the increase in CO2 concentration and saturation is seen only beyond 450 micro per liter. Thus, current availability of CO2 levels is limiting to C3 plants. The facts that C3 plants respond to higher CO2 concentration by showing increased rate of photosynthesis leading to higher productivity has been used for some greenhouse crops such as tomatoes and bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that leads to higher yields. Temperature. The dark reaction being enzymatic are temperature controlled, though the light reactions are also temperature sensitive that are affected to much lesser extent. C4 plants respond to higher temperature and so higher rate of photosynthesis, while C3 plants have a much far lower temperature optimum. The temperature optimum for photosynthesis of different plants also depend on the habit that they are adapted to. Tropical plants have higher temperature optimum than the plants adapted to the temperate climates. Water. Even though water is one of the reactants in the light reaction, the effect of water as factor is more through its effect on the plant rather than directly on photosynthesis. Water stress causes the stomata to close hence reducing the CO2 availability. Besides, water stress also makes leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well. So here's a summary. Green plants make their own food by photosynthesis. During this process, the carbon dioxide from the atmosphere is taken in by leaves through stomata and is used for making carbohydrates, principally glucose and starch. Photosynthesis takes place only in the green parts of the plants, mainly in the leaves. Within leaves, the mesophyll cells have a large number of chloroplasts, whereas the cells for CO2 fixation. With the chloroplasts, the membrane are site for light reaction, while the chemosynthetic pathway occurs in stroma. Photosynthesis has two stages: the light reaction and the carbon fixing reaction. The reaction is light reaction. The light energy is absorbed by the pigment present in the antenna and funneled to special chlorophyll A molecules called reaction center chlorophyll. These are two photosystems, PS first and PS second. PS first has 700 mm absorbing chlorophyll. A PS 700 molecule its reaction center, while PS second has a P680 reaction center that absorbs red light at 680 nm. After absorbing light, electrons are excited and then transferred through PS second and PS first and finally to NAD form an NADH. During this process, a proton gradient is created across the membrane of thylakoid. The breakdown of photons gradient due to movement of through F0 particle of ATP phase ATPase enzyme releases enough energy for synthesis of ATP. Splitting of water molecules is associated with PS second, resulting in the release of O2 protons and transfer of energy to PS second. In the carbon fixation cycle, CO2 is added by the enzyme RuBSCO to a 5 carbon compound RuBP that is converted to 2 molecules 3 carbon PZA is converted to sugar by Calvin cycle and RuBP is regenerated. During this process, ATP and NADPH are synthesized in the light reaction or utilize RuBSCO to catalyze a wasteful oxygenation reaction in C3 plants. Photorespiration. Some tropical plants host a special type of photosynthesis called C4 pathway. It is the first product of CO2 fixation that takes place in the mesophyll is a 4 carbon compound. In the bundle sheath cells, the Calvin pathway is carried out for the synthesis of carbohydrates. Here the chapter is finished.